Okay.
Hello.
Hello.
Yeah.
Yeah, welcome, sir.
Okay. Good.
Yeah, yeah. So,
So, can you hear me? I can hear you.
yes, we can hear you
I
also.
think
Yes.
I'm comfortable without earphone also. So, is that okay? Or?
Yeah, that's fine. Yeah, that's fine. That's fine. Yes. Okay, we'll start now. Uh,
Yeah, fine.
yeah.
So
Good
shall I
afternoon,
introduce?
everyone. Yeah, good
Yeah.
afternoon, everyone. Welcome to the today's FTP session. I request Dr. Sainthan to chair this session and introduce the speaker.
Thank you so much, Siddharthan sir. So it gives me immense pleasure to invite Dr. Ahmad Ali. Right now, he's posted in the University of Mumbai, the Department of Life Sciences. He's serving as assistant professor over there. So I am knowing him since 2010 when I was a research scholar, and my guide invited him as a resource person in the refresher course over there. So it is similar to that event itself. So it's a really great, what do you say? It's very exciting for me to invite him. And I will just, in very brief, I'll introduce him to all of our department faculty members. And he had pursued his doctoral research from the University of Mumbai, and he completed his degree in 2007. And after that, he has worked in the University of Mumbai as a teacher. And after that, he joined NIPER, National Institute of Pharmaceutical Education and Research in Hajipur. And I got to know him when he was there. And after that, after working for 2 years, he went to University of Mumbai to join as full-time assistant professor in the department. So he has various achievements. So till date, he has completed the four research projects funded by national bodies, and currently he's working on one research project. So his major area of research, if I'm not wrong, is biochemistry, as I saw from his publication and as I interacted with him before. So, and he's member of various national bodies like Society of Biological Chemists India, Indian Science Congress Association, European Federation of Biotechnology, Science Advisory Board of India, to list a few. He's member of advisory committee of various national and international conferences, and he has organized as well. So, and he has more than 23 publications in national and international journals, as well as he has 8 book chapters credited to his name. So, with this, I invite Dr. Ahmad Ali to take this, what you say, not the dais, but I will invite him to deliver his lecture. Thank you so much for coming, sir. Thank you so much.
Thank you, Dr. Santan. It's nice meeting after a long time, and thank you for your introduction and kind words. Uh, I'm also thankful to the university and all the officials for giving me this opportunity to interact with you, especially Dr. Siddharthan, who has been coordinating with me since many days. So I have been looking forward to interact with this, and as I have been briefed that it's a part of FDP. So I'll talk about my research work which is going on in my lab. The main theme. Okay. So the main theme if my in my lab is glycation. So I'll talk about the basic facts about those, and then I'll briefly introduce what we are doing in my lab. I'll not be talking about other projects which we are doing in the lab. So may I start now? Dr.
Yes, yes, go
Siddharthan.
ahead. Yeah, go ahead. So you can share your screen, share your presentation. Now it's clear. Yes.
Thank you. Okay, sorry for that inconvenience. Okay, so as you may be aware that the theme of the title of the presentation is glycation, which is an important biochemical link between diabetes and secondary complications. So as you can see here, I have taken this diagram where people talk about advanced glycation and products and skin diseases and aging. So these are some popular websites which show this kind of thing, and they market their medicines using glycation as that. This is another one which show that sugar can give you wrinkles, means excess amount of sugar can lead to the wrinkles. Am I audible? Hello.
Yes, sir. You are audible.
Sir, can you
Yeah,
yes,
make
it's
this
audible.
full screen? Yeah.
Yeah, it's
Pardon?
audible.
Voice breaking, sir.
Am I audible or it's?
It's audible. It's audible.
Yeah.
Yeah,
But there
you are
is some
audible.
breaking, it seems. So
Okay.
now it's
Please do let me know
Clear.
if,
In my case, it is clear. Our volume is clear.
clear.
Yeah,
Audio is clear.
yeah, if I am not audible, please do let me know if there is any.
So just please shift F5, sir, so that the entire screen we can see, sir. Yeah,
Okay.
shift F5.
Okay.
Yes, now it's
Yeah, sure, sir. Yeah.
So these are some. I mentioned that these are some website images from popular, not scientific ones, where they are marketing their products as anti-glycating and antioxidants to say that okay, this will solve your aging and wrinkle problems, using glycation as one of the concepts behind that. I'll briefly also talk about diabetes, because glycation is involved in diabetes. So I'll just give a few uh, points on diabetes. Although all of us are aware, it has become a common talk in the field of science as well as government sector and health sector. So this is a very common slide, and it can be easily understood, like what are the symptoms which are associated with diabetes and what are the complications. And so basically I'll be talking about glycation. So we all know that uh, every organism has many biomolecules, and they are the driving force. So these molecules, they perform their function not in isolation, but they interact with each other, and that interaction is very important, which leads to some of the very crucial these things for like generation of energy and raw material required to carry out the work. Okay. So if there is any kind of impairment in this biochemical reaction, we can see the consequences, and the consequences may be toxic accumulation of reaction products, damage due to the oxidation, deficiency of an enzyme or protein, or some over stimulation of certain reaction pathways. So there are many impairments in the biochemical interactions and pathways, but I'll concentrate on glycation, which is in very simple terms a non-enzymatic amino-carbonyl reaction between sugars and proteins, lipids, and nucleic acids. We all know glucose is one of the very important metabolites for our body, and it comes as a result of metabolism of carbohydrate or degradation of carbohydrate. So it's instant source of energy, and it becomes it provides energy as well as some precursors for other metabolic processes. So the metabolism of glucose has to be very balanced. Any kind of impairment or fluctuation will lead to a disease conditions or a combination of disease conditions. Most commonly, the two conditions which are associated with glucose availability and utilization are hypoglycemia and hyperglycemia. So these two impairments or fluctuations of glucose metabolism are actually involved in diabetes. So it has, as we all know, that it has become a major public health concern globally, and for our country also, because the recent report suggests that out of every 100 diabetic people, 30 people live in India, and as we can see from we have a fourfold increase in the number of diabetics from 1982 to in 2014, and 50% of this population lives in these countries: India, China, USA, Brazil, and somewhat except US, some of these are developing countries also. We can see that this is the projection. In 1995 we had almost 100 million, and in 600 by 2045 we may have more than 600 million diabetics in the. So the diabetes, the process of this this disorder, it originates from or manifests the dysregulation of glucose metabolism, and it is one of the most common metabolic disorders among others. So there are two types. I'll not go into detail of this, type 1 and type 2. Because of the result of inability of the pancreas to produce insulin, we see type 1, and because of the inability of the cells to utilize the produced insulin, we see the onset of type 2, that is more common. So as a result of diabetes, I said that diabetes is one disease condition which leads to many complications, and it can be seen in, or it can lead to obesity, nephropathy, hypertension, dyslipidemia, non-alcoholic fatty liver disease, and systematic inflammation. So based on the literature information available, there are multiple hypotheses on the origin of complications in diabetes. You can see carbonyl stress, you can see high bone mineral density, advanced glycation end products hypothesis, oxidative stress, and so on. So these are another very common, like when if you see the major root causes of diabetes, that has been summarized like inflammation, toxicity, oxidation, and insulin resistance, of course, and glycation is also. So this, with this slide, I'll summarize the diabetes concept. So diabetes management can be done by any of this, by activation of AMPK, inhibition of glycosylation of hemoglobin and other proteins, modulation of glucose uptake, inhibition of aldose pathway, AGEs inhibition, enhancement of insulin secretion. So these are the main, when a drug is developed, these are the major ways by which diabetes can be controlled. But we have been talking about many other concepts, like, okay, drug is one thing, but we need to change our lifestyle and other things. So major symptoms or major symptoms associated with diabetes are hyperglycemia and hyperlipidemia. These two conditions, what they do, they induce inflammatory immune responses like glucose oxidation, auto-oxidation, induction of polyol pathway, and synthesis of prostaglandins, thromboxanes, and glycation of proteins, which ultimately lead to the generation of oxidative stress, and finally it impairs the functioning of antioxidant defense system. So as a result of prolonged hyperglycemia, it results in deterioration of blood vessels, causes damage to vital body organs like kidney, nervous system, eyes, heart. So when we classify, or when we subgroup this kind of disease conditions, we see two conditions: macrovascular, where we see heart attack, stroke, and peripheral vascular disease, and microvascular, retinopathy, neuropathy, and nephropathy. As I mentioned that I'll not be going into detail of this because I'm trying to bring to glycation. So I have started with molecular interactions and diabetes, and then I have come to this hyperglycemia, which is a condition that is known because of accumulation of glucose, which ultimately leads to accumulation of undesired products due to excess amount of glucose present in the body. So if you see diabetes, the two common features which is noticed is hyperglycemia and glucotoxicity. Glucotoxicity means an abnormal level of glucose accumulating in the body. So based on all these concepts and recent informations, the modern theory of diabetes says or is centered around oxidative stress and glycation. So if we have diabetic hyperglycemia, we see many kind of protein modifications. We have protein carbonylation, protein nitration, succination. Means when there is an accumulation of glucose in the body, the glucose is involved directly or indirectly in the modification of the proteins, and finally lead to some kind of loss of function of those modified proteins or hyperactivity of those proteins. So as you can see in the slide, there is also a protein glycation, and the information or the research suggests that it's one of the most important or significant process of protein modification, which is actually a direct manifestation of reductive nature of glucose. We all know that glucose is a very highly reducing sugar. So the process of glycation can be defined as a spontaneous non-enzymatic amino-carbonyl reaction between sugars and long-lived proteins, lipids, and nucleic acids. As you may notice, that a term has been introduced in this, that is non-enzymatic, because we are also aware that protein glycosylation takes place in ER as a post-translational modification. So that is under the control of enzymes. But this process of glycation, which is also known as non-enzymatic glycosylation, because of non-involvement of enzymes. And another specific thing about it is that this reaction is between the amino group and carbonyl group of the proteins and sugars respectively. It's also one of the post-translational modifications, and it is an irreversible process. I have shown a cartoon. You can see that it's a blood supply where you have sugars, albumin, hemoglobin, the amino group of the proteins which are present on the basic amino acids like lysine and arginine, and the free N-terminal amino acids, they are modified by this glucose. As a result, there is a formation of SIF base and some other products, and finally you see that you see the proteins which are known as, in general terms, glycated hemoglobin or glycated proteins or glycated albumin. So this is a more detailed diagram which shows that how glucose and other processes are involved in the formation of AGEs. I'll come to that AGEs later on, but you can see that this process takes place in several weeks or starting from several hours. So another kind, you can see that what are the impact of this. I'll discuss later on also. So the process of glycation can be divided into 2 or 3 stages. Somewhere it is said that 2 stages, somewhere it is mentioned 3 stages. We'll stick to this 3 stage concept. So in the early stage, there is the interaction reaction starts with the interaction between the aldehyde group of glucose, which is also responsible for the reducing property of this sugar, and the amino group of lysine and arginine or N-terminal amino group of the proteins. That leads to this aldehyde and primary amino group interaction leads to the formation of a SIF-based adduct, which undergoes further modification to form early glycation products, which are also known as Amadori products or Hens products, by the because of the name is because of the person who discovered this Amadori and Hens product. So this is the common this is the scheme. You can see in the early the sugar and proteins interact, shift bases are formed, Amadori products are formed, and then in the next stage they further go modification and dicarbonyl compounds are formed, and in the late stage, advanced glycation and products are formed. So intermediate state, as I mentioned, that shift bases and amardori products undergo some kind of modifications and lead to some of the very reactive dicarbonyls, which are actually involved in the most of the complications of glycation. These are methyl glycol, and deoxyglucosone. In the late stage, these amardori products and dicarbonyls, they further go uh, undergo chemical reactions or rearrangements like oxidation, reductions, and hydration, and it are involved in the formation of cross-linking of the proteins or aggregation of the proteins, and that completes the third stage of the uh, or the last stage of this glycation. So finally, what we see is the brownish products, and uh, this deleterious or a combination of heterogeneous compounds are known as advanced glycation products, or in short, AGs. So if we are under the assumption that this glycation happens in the hyperglycemic condition, that is not true. The glycation continuously happens. Body has a mechanism to control them. So this, if that is not controlled, this glycation products slowly accumulate in the body, and the situation gets worsened in the hyperglycemic conditions, where you have more sugars. So this is a cartoon which shows that, okay, if you have normal level of sugars, the glycated albumin is normal. But if you have a high amount of glucose or hyperglycemic condition, then you can see that the glycated albumin quantity has increased. That is one of the parameter to measure it clinically. We can see here that if we want to assess the diabetic conditions, because I told you that diabetes can be measured, as we know that diabetes can be measured by various serological and other tests, but one of the tests which is quite reliable is the measurement of glycated hemoglobin, that gives an idea about the abnormal level of sugar from 1 to 3 months. On the other hand, if we measure the amount of glycated albumin in the serum, it gives about the hyperglycemic condition for a few weeks, like from 2 to 4 weeks. We need to be careful because this the level of these glycated proteins are altered under some disease conditions like nephrosis, cirrhosis, anemia, and renal failure. Okay. Some of us might have heard of this Maillard reaction. It was Louis Camille Maillard who in 1912 was working on amino acid synthesis. When he saw that when amino acids react with sugars, there was a color change in the browning to the brownish, and since then this process is known as, and the food industry is very well familiar with this Maillard reaction. They are worried about it, or they introduced this. There are some controversies also that food industries introduce this glycation products to increase the taste, or to enhance the color by having this brownish color. As we also see that in our day-to-day cooking, we like deep fried or brown products. So something about, I'll talk something about or in detail about ages. So they are very heterogeneous group of molecules. There are three routes by which formation of ages take place: auto-oxidation of sugars, Amadori reagents, and SIF products, SIF adducts. So these are all internal sources of generation of ages, or what we call endogenous sources of ages. But we have another very good source of this ages, that is known as exogenous or ingested food. So As I was mentioned about the three pathways of the formation of ages. So, am I audible, or if, am I going fast, or something?
No, no,
I
no. It's
would like to
clear.
have
It's
some.
audible and it's fine.
It is. I mean, it's okay. All clear. I mean, this is.
It's clear. The speed is clear. Yes. It's fine.
Yeah. Okay. So I hope I'm not going very fast because.
No, no. We are understanding clearly.
Yeah. Thank you. So please do stop me whenever if there is any clarity. So these ages, they have some common feature that they act as cross-linking agents and they are fluorescent. So on the basis of these properties, they have been divided into three groups, basically fluorescent, non-fluorescent, and cross-linking agents. Some of the examples of these advanced glycation products are carboxymethyl lysine, carboxyethyl lysine, perylene, and they can be characterized by and measured by different issues. I am showing this diagram because to show the structure of this pentosidine, carboxymethyl lysine, and perylene, which are produced as a result of the late stage of the glycation. Okay, so what are the factors which affect age production? Internally, it's the presence of oxygen or reactive oxygen species or redox active transition metals. They all they all increase the production of this ages. Diet and smoking also influence the production of ages. Now, how do these ages play role in diabetes? So as we can see from this diagram, if someone is having high glucose level, that induces the generation of mitochondrial reactive oxygen species that leads to oxidative stress and beta cell dysfunction. Through ages, when glucose is converted to through glycation leads to production of ages. This age binds to a receptor that has been given the name like receptors for ages, that leads to I'll talk in the next slide, that leads to again ROS production, which leads to NF kappa B activation and inflammation, ultimately leading to insulin resistance. And both these conditions, beta cell dysfunction and insulin resistance, are involved in the onset of diabetes, or they manifest. So if someone wants to measure the presence of advanced glycation in products, some of the major or the common sites are, one of this is like we can measure in blood, another is in skin, urine is another source of this or can be shown to have cartilage, as well as eye lens. I was talking about this glycated hemoglobin. So glucose circulates in the blood circulation, where it encounters albumin or hemoglobin or other proteins. If you remember, when I defined glycation, it mentioned that the interaction is between the carbonyl group of the sugar and the amino group of the long-lived proteins. In one of the diagrams also, I mentioned that it takes several days or several weeks actually for this glycation to happen. And we know that hemoglobin and albumin have quite longer half-lives. Collagen is another protein. So the high level of glucose, when it goes through circulation, it interacts with hemoglobin and binds to hemoglobin, leading to the formation of glycated hemoglobin. So higher the level of, or if there is continuous increase in the level of continuous, the condition is sustained hyperglycemia, there will be an continuous increase in the level of hemoglobin. And if someone someone might have noticed it, that doctors do prescribe this test for, it's slightly costly, but doctors do prescribe to get information about long-term diabetic conditions, because sometimes fasting this blood glucose levels don't give an idea. So, but again, this also has some controversy, but it is one of the most accepted parameter by which diabetic can be measured. So as you can see that this cartoon shows that hemoglobin is there in the RBCs, and this glucose enters and interacts with the hemoglobin and leads to the glycated hemoglobin, and ultimately this hemoglobin loses its function. So what are the complications which arise due to, or where are the complications, what are the complications where glycation is involved majorly? Diabetes mellitus is one of them, of course. We see aging, aging is skin aging and other processes. The major root will be the formation of ROS, Alzheimer's disease, Parkinson's disease. These are basically aggregation based disease. Cataracts, we see there we see the accumulation of sorbitol, as where high level of glucose is there and glucose is converted to sorbitol and fructose, that is known as polyol pathway. So cataract is another complications which arises due to glycation. We see it in retinopathy, arteriosclerosis, because of the deposition of this glucose and other products, and increased susceptibility to the. So this is this diagram again shows that glycation on the where it can be involved. So if we at molecular level, if we see what glycation does, it basically the glycation or the glycation products are basically involved in the process precipitation of the proteins or cross-linking of the proteins, aggregation of the proteins. So all this precipitation, cross-linking and aggregation, they lead to the loss of function of these proteins, and then these proteins, then this modified proteins are degraded by the proteasome systems. So we have also shown that ages lead to the production of reactive oxygen species and reactive nitrogen species, and this glycation adapts as well as reactive species. They also lead to the damage to the lipids as well as the strand breakage of DNA. So basically the glycation, the glycation products, they alter the structure of the biomolecules. That's why in the first, in the one of the first slide, I mentioned that one of the major consequences of loss of this or impairment of biomolecular interaction is glycation. So as we can see that this interaction leads to damage or alteration of the structure of the proteins and other biomolecules. So we have some intrinsic system to control this toxicity of glycation. We have aldehyde reductase, glyoxylase, and amidoriases. We have proteasome systems which degrades age-modified proteins. We have processing mechanisms through age receptors, macrophage systems which dispose peptides, and we have some drugs also. One of the major drugs till recent time was aminoguanidine, but again it has its own side effects. So we are turning to the natural products like vitamins, pyridoxine especially, and we have anti-glycating agents and antioxidants. So the modern theory suggests that if some molecules which act as antioxidants can also act as anti-glycating agents, because ultimately one of the major consequences of glycation is generation of free radicals. Okay, so how can we reduce the level of ages in our body? One of the most important is that we consume less amount of sugar. We eat vegetables and fruits raw, boiled or steamed. We don't overcook. Limit our consumption of processed and brown foods. Cook meats at low and or a slower rate. Avoid high fructose corn syrup. Drink lots of water. Supplement with pyridoxamine and stop smoking, because all these things lead to the ages. So one of the recent advances in last 10 years is the discovery of these receptors which belong to immunoglobulin family. They are known as RAGEs because they were discovered to bind to ages, but they bind to other proteins also, and binding of this age to RAGE and other ligands lead to a cascade of signaling pathways. So these ages are found in many of, or most of the cells, like endothelial cells, smooth muscle cells, cells of the immune system. So what do ages do? They bind to these receptors, spawn reactive oxygen species, and lead to a signaling cascade of the reactions, which ultimately lead to the inflammatory responses. Okay, so ages have been shown to be involved in, and lots of research is taking place at this RAGE and other ligand uh, complex. So they are involved in diabetes-related chronic inflammatory diseases such as atherosclerosis, asthma, arthritis. So because of this, some drugs which are being developed, they target this RAGE actually. So so all these processes, all these disease conditions are because of the activation of NF-kappa B, and this NF-kappa B it controls or regulates the several of the genes which are involved in the. So you can see in this diagram age and other ligands, they bind to RAGE, that leads to some kind of signaling and activation of some of these downstream signaling proteins, and that leads to activation of NF-kappa B, increased transcription of TGF beta, PGDF, and all those things. So ultimately, now how can we prevent the formation of ages in the body? Or in the previous slide, or one of the slides, I said what we can do as, but when it comes to development of the drug, how can we what have been the strategies developed so far to control? It's a very difficult process to control because it involves several stages, and one of the stages may be skipped. So preventing the formation of ages by inhibitors like ascorbic acid, aspirin, and metformin, they are all antioxidants. Deglycation of CYP bases and Amadori products by hydrazines. These hydrazines, they scavenge the or inhibit the formation of the CYP bases. Then reversal of age-induced modifications like cross-links with the help of phenyl thiazolium bromide, and prevention of the body from ill effects of age formation by using natural compounds like resveratrol and curcumin. This is a scheme where we show that what are the strategies. So if you can see that there are various strategies to prevent. You have A, B, C1, C2, C1, C2, E1. So they are at different levels, and different molecules have been designed to work at this different metabolic processes. So some of them are metal and chelators, some of them are antioxidants, some of them are hydrazines. So one of the intrinsic enzyme system which I mentioned in one of the slides, I'll show you this, that there are 3 enzyme systems which have been identified in the body, and some work has been done on these enzymes also, and people are trying to characterize these enzymes, but not much work has been done on these enzymes, unfortunately. These are oxidases, they belong to oxidases, isomerases, and kinases. You can see here in this diagram, this is the glycated glycated protein or glycated product. So you have amidoriases, and you can remove this, then you get ketoaldose. Then you have deglycases, then also you get you remove this you break this shift base formation and remove this, then you get aldose, and then you have ketoamine kinase, so you get this. So these are three groups of the enzymes which are present in our body. They work to prevent the formation, as I mentioned in one of the slides, that we always have glucose level in the body, and they interact with the albumin and hemoglobin and other long-lived proteins. The resultant of those interactions are taken care of by this enzymes as well as antioxidant systems in our body. So my focus was basically the biochemistry of glycation. So what methods have been used to analyze glycation uh, products, or measure the glycation products, and some drugs which can act as the anti-glycating agents. So one of the very preliminary method is NBT assay, where we measure the fructosamines. It's all this is a colorimetric method. Then we have a DNPH assay method, which measures the last stage of this glycation, that is protein carbonyl formation. Then we have DTNB, that is detection of thiol group in Amadori products. Then we have then we can use some assays to measure the glycation induced aggregation of the proteins by Congo red and thioflavin, because glycation also induces or glycation advanced ages induce aggregation of the proteins, which lead to formation of amyloid cross beta structures. That's how they are involved in Alzheimer's and Parkinson's diseases. And there is another T-bars method, that is again for the measurement of lipid peroxidation products, or lipid oxidation products, or and some of these analytical methods are ELISA to measure fluorescent and non-fluorescent ages. Then we have HPLC, then we have LCMS, UV visible, and then we have boron affinity chromatography. So some of these major methods are being used to measure both glycation products and the anti-glycating activity or potential of any product. So in what we have been doing, we are working on several aspects of this glycation. So we have incubated, basically we are working on in vitro model, where we incubate the proteins with various glycating agents like glucose, fructose, and methyl glyoxyl or glyoxyl, and so on. And then we measure the level of or the amount of glycation products by some of the methods which I have mentioned in the previous slide, and some of the test compounds which we have used to measure how do these products prevent the formation, or whether they are able to prevent the formation of ages or not. So in this we have taken three natural compounds like thymine, curcumin, and thymoquinone. I have taken one of the compound that has also been a major interest in our lab, that since last 1 decade or so, there has been an increased intake of artificial sweeteners. So our interest was that whether these artificial sweeteners are also involved in the glycation process or not. So we work with several of the FDA approved artificial sweeteners like saccharin, sucralose, aspartame, neotame, and aspartame, this asulfame potassium. I'll be basically talking about asulfame potassium here because it showed quite interesting result here. So some of the assays which we have done in our lab are browning. So as I mentioned in one of the slides, that the glycation is also known as browning event, or it's a Maillard reaction, because Maillard showed that when amino acids and sugars interacts browning, the color changes to yellowish brown. And we have measured fructosamine by NBT method, carbonyl content, total ages by spectrophlorimetry, aggregation by congruent thioflavine. Then we have measured glycosylated damage. Then we have done some quenching studies. Then we have done some structural alteration like electron microscopy or DLS or CD also. I'll show some of the results. We have also done some docking studies here. So starting with the thymine, that vitamin B group member. So I hope you can see that the middle bar is shows that BSA plus Mg, which means that this is the glycation system, and we can see that after incubation for 3 or 4 weeks or 1 week, there is an increase in the browning, an increase in the fructosamine content, as well as increase in the carbonyl content. When we add thymine, we could show that there is reduced browning, slightly decrease fructosamine, and a significant decrease in the carbonyl content. On the basis of this three, we could show that thymine has significant property in reducing the. Again, we worked with curcumin. Curcumin also we could not see much of the and browning has not been recent. I mean, in recent times browning. Sometimes browning happens and sometimes doesn't happen, but it doesn't mean that there is no formation of ages. But still, people do take this as a preliminary measurement. So what we could show that curcumin significantly could reduce fructosamine content, and this carbonyl content is the consequence of both decrease in the glycation as well as antioxidant property of the molecule. We also did some kind of quenching study with the BSA, where we could show that this is as we go on increasing the concentration of curcumin, there is increased quenching of the BSA. So that shows that curcumin interacts with BSA. So this is a major phytoconstituent of black cumin or Nigella sativa. We have also done quite a lot of work on this thymoquinone. So we used 2 different concentrations of this thymoquinone in this study, 10 micromolar and 20 micromolar, and all these studies like browning measurements and browning measurement, fructosamine and carbonyl, shows that thymoquinone is has very good potential in reducing the or preventing the formation of glycation products. Then we did the congruent and thioflavine T assays. So as you can say that thymoquinone has prevented the formation of amyloid beta structures, which can be seen by both these. So as you can see that this is the aggregation level of the BSA glucose, and this is the aggregation level of proteins in the presence of glycated proteins in the presence of thymoquinone at different concentration levels. So then we measure the total amount of ages. As I mentioned in one of the slides, that some of the ages are fluorescent and some of the ages are non-fluorescent. So one of the parameters can be used by measuring the fluorescent ages, what we call as total ages, although it is not total ages because we are not measuring non-fluorescent ages, but it gives an idea that how much ages have been produced. So you can see that the 20 micromolar of this thymoquinone has significantly reduced the amount of fluorescent ages Then again we did quenching studies that, to show that whether the interaction is between the thymoquinone and BSA. So we could go up to 10 micromolar and we could show that okay, the quenching increased with increased concentration of thymoquinone. This diagram shows that we have used PBr 322 here. And why we have used PBr 322? Because PBr 322 plasmid can go into different conformations like supercoiled, circular, and linear. In the first lane, this is the control, and in the second lane, that is just thymoquinone. And in the third lane, we can show here that the introduction of glycation system. Here we have used glycation system as lysine and Mg, and we have introduced FeCl3. FeCl3 is a catalyst which catalyzes the formation of free radicals by a reaction, well-known reaction mechanism known as Fenton reaction. So we have used. So in the lane 3, we could clearly show that the supercoiled DNA has converted to what you can see that circular DNA. But the moment we add thymoquinone, we can show that this damage to DNA has been prevented by this thymoquinone. So we can conclude from this that thymoquinone is quite able to prevent the glycoxidative damage of DNA. So now I'm coming to asulfame, and when we started this work, we were thinking that asulfame or some of these sweeteners may be involved in increasing the formation of advanced glycation products. So, but strangely, we could find that asulfame, one of the sweeteners, could actually prevent the formation of glycation products. So we can show here that this is the BSA, this is the glycated protein level, and this is the glycated protein level in the presence of asulfame K. So same way, there is a reduction in the production of early glycation products, and there is a reduction in the production of late glycation products. So with this initial informations, we went ahead and we we checked whether this glycation induced aggregation is being prevented by asulfame K or not. So we could show that okay, this aggregation is being by both the Congo-Red and thioflavin T acids. We could show that asulfame is efficient enough to prevent the formation of thioflavin aggregated products. So again, with the total ages, we can show here that there is a significant decrease in the production of total ages. Then we checked with whether this asulfame is able to prevent thermal aggregation or not. So we could show here that after 6 hours, we can show here that BSA and glucose plus asulfame was quite efficient in reducing the level of thermally aggregated proteins. So as we did with thymoquinone, we also did with asulfame potassium. In this we can see in this lane 2, we can show here that the plasmid has been converted to supercoiled and linear form, which has been prevented in the presence of asulfame potassium. So again we did a quenching study, and with this we could show that this quenching study is very important for drug development to show that whether there is an interaction between the ligand and protein or not. So we could show that with increasing concentration of this asulfame, there is a decrease in the intensity, means increased quenching. So we wanted to confirm whether that aggregation is uh, which we have measured with Congo red and thioflavin T can be actually is actually happening or not. So we use transmission electron microscopy. So we can show here that BSA when it is there after incubation for 4 weeks. So we can show here that glucose induced heavy aggregation in BSA having 200 to 5000 2000 to 500 nanometer Hughes patches of electron dense structures. On the other hand, when asulfame was added in the presence for this, the particle size reduced to 50 to 300 nanometer, which is similar to what we have with the BSA. So with this electron microscopy image also, we could show that asulfame is able to prevent the aggregation. Then we did the DLS study to measure the particle size, and we could show that asulfame is again reducing the size of the particles. So then we did a molecular docking study where we wanted to know whether the binding site for BSA, binding sulfide, glucose, and asulfame, is there any overlap or not? So we could show here that BSA binds to glucose binds to BSA with an affinity to of this molecule on this serine, histidine, and proline and arginine 40 register. And it was realized that, and by this docking study, that asulfame also showed comparative binding affinity of almost the same energy level on the same residues. So we can hypothesize here that whatever anti-glycation property which we have shown here is because of the mostly is because of the interaction between the BSA and asulfame, and the overlap between or a competition between the binding site for glucose. And so we did some other kind of studies also. I'll just quickly wind up now. BSA has one of the activities that is known as intrinsic activity, that is known as esterase activity. So we could show that when BSA was glycated, the level of this goes down quite significantly. But when we added asulfame, the esterase activity is restored. So it means that asulfame K prevents the structural alteration of the protein because it prevents the glycation of the protein. Okay, so I'm coming to discussion like the thymine and curcumin could decrease browning and protein oxidation. This thymoquinone and asulfame, they were better in reducing the early and late glycation products. So, and the glycation induced, they also reverse the oxidative damage of DNA. And this quenching studies of BSA with curcumin, thymoquinone, and asulfame showed interaction with them. So finally, to conclude this some of these natural products can be quite efficiently used as anti-glycating agent, and the added advantage is that most of these natural compounds are also antioxidants. So it fits into our modern hypothesis of diabetes. We say that oxidative stress and glycation. So these are suitable candidates. They can be used to prevent the glycation-induced aggregation. But we need to analyze what kind of interaction is happening, and so this work has been carried by my PhD students and MSc project students. I received funds from UGC, RSSBI, and University of Mumbai, and we have DST infrastructure. We have some collaborators from NIRH and NIV Pune. So with this, I'm thankful to you.
Thank you very much, Dr. Ahmad Ali.
Yeah.
So is there any
Should I
questions?
stop presenting?
Yeah, stop the slides and you can. Are there any questions?
So if there is any question from people.
Yes, Suma.
Thank you, Dr. Ali. Can you hear me?
Yeah, I can. Yes.
Okay, one small doubt. So, are you trying to indicate that SLP can competitively inhibit with glucose for the binding?
Pardon, can you please repeat again?
SLP, two questions I have. One is SLP will compete with glucose for binding.
That is what our docking study shows that.
Okay. Second question is, SLP being an artificial sweetener, won't it have any other side effect, or it is foolproof? I mean, safe?
As far as acceptance is concerned, SLPM has been accepted by or approved by FDA, and it is safe. But again, especially in USA and European countries, there is a lot of debate going on about the safety of this artificial sugars, and there are reports where people have suggested that some of these sweeteners lead to obesity.
Okay, yeah, I had read on those lines. Yeah.
But still, FDA has not banned the. I think one of the sweetener has been saccharin has been, because of this carcinogenic activity, has been withdrawn from the market. But FDA is waiting for substantial proof to show that this. Actually, I was also my aim was also to show that, because now most of the beverages and food products have this sweeteners now, and even those who are diabetics, they are advised to take artificial sweeteners.
Yeah.
So the consumption level of sweeteners has been for some people is more than the natural sugars. So
okay, so thank you.
Yeah.
Dr. Ali sir, good evening, sir.
Good evening.
Sir, is artificial sweet is more dangerous than the sugar which we use for beverages?
I think the question is again that safety level. We have not been, but one thing we can. A wonderful molecule like glucose is still, being a biochemist, I always find it very difficult to accept that this glucose can be so dangerous. But if a natural molecule which has been accepted and adopted by our body can be dangerous, so if using the same theory, this artificial, if the intake of this also increases, and it's going on increasing by every day, so this also may lead to major consequences.
Thank you, sir.
But people are trying to do some kind of lab studies on the animals and human studies, like whether. But so far we have not been able to accept this obesity and this thing. We have not been able to show that these sweeteners are actually involved in some kind of disease conditions or not. But of course they may be harmful if they are consumed like sugar.
Yeah.
Hope I could.
Thank you.
Hello, sir. Good evening.
Yes.
Yeah, I have one general question. Does habit of drinking cold water reduces the fat, and what
Pardon,
is the effect? Can you hear me, sir?
yeah, I can. Can you please a bit be little?
Yeah, I am asking. Hello. Does the
Yeah.
habit of drinking cold water reduces the fat? In that case, what is the effect on diabetes?
If we take more amount of
Cold water.
Cold water, if you
cold water,
yeah, general.
ideally, I mean, it's very difficult to say that whether the cold water will have any kind of effect or not. But if you go by natural theory, cold water will be harmful. But per se, whether it is harmful for diabetes or glycation, it's very difficult to say at this stage. Or I have not come across any report which.
Okay.
But I think all of us are aware that okay, this consumption of cold water, I mean below normal temperature, is not a healthy practice. Either we should drink a normal water or slightly warm water in the morning. There are many doctors who are suggesting that we should drink warm water or this thing. But at least cold water is not very good for health. But whether it is bad for diabetes or not, I don't know exactly. Yeah.
Thank you.
Yes.
Siddharth, can I ask one more question?
Yeah, you ask.
So Dr. Ali, you had mentioned about those receptors for ages, right?
Yes.
Do they have, I mean, what is the normal function in the body, or does it have only this function? If it doesn't have any important function, can we think of having inhibitors for those receptors to prevent this?
People are designing drugs for those, using those rages as because they can be very good candidate for developing ligands on the. They are normal receptors. They are required for our body function. I could not show another diagram where we have shown there are S100 proteins and other proteins which use RAGE as the receptor. Okay, so they are required for normal. If you remember the slide, it is required for the NF kappa B activation, and NF kappa B activation is required for our immune function. All inflammatory responses are through NF kappa B, and so it's normally it is required. But what I showed, that if there is an accumulation of ages, so they go and bind to rages and those. So the inflammatory, normally also we have inflammatory responses in our body. They are good for some kind of infections and all those things. But if there is an increased amount of ages and in our body, then it will be harmful. We don't want to have those inflammatory responses and reactive oxygen species in our body. So those who are diabetic, they are always having this high level of glycation products and oxidative stress. So for them, people are trying whether having looking at this RAGE age complex and reducing the RAGE signaling will be useful for them or not.
Okay. The inflammatory bodies which you told about, do they accumulate forever, or is there any reversion?
Once Amadori is formed, it leads to formation of an advanced glycation products.
Okay.
So the best way is to prevent the formation of SIF base, the first stage.
So where does this glycation generally happen? In the blood plasma only, or so? In that case, all these compounds like curcumin or ASK and all, will it be targeted to those tissues where the glycation is happening?
Glycation is like when it happens everywhere.
Everywhere in the okay.
Because glucose is there everywhere.
Okay.
Glucose can go anywhere.
Okay.
One of the most one. So, but nature, because I mean it may be a nature's design also. Only those proteins which have long half-life, they get affected.
Okay.
For example, albumin has a longer half life, like this hemoglobin has, and one of the worst affected protein which in this is collagen is there.
Okay.
Then I can ask one question.
Yeah, yes.
Sir, does the protein complexity depends on glycosylation?
Which one?
Protein complexity, does it depend on glycosylation? If a protein is simple and complex, do the glycosylation vary?
Uh, I'm unable to. Sorry, I could not hear your question. Can you please repeat it, or
Okay.
can you?
Okay. Sir, the complexity of the protein, does it influence glycosylation?
Yes. So that's a natural product process which happens in the cells post translational modification, where proteins are most of the proteins are glycosylated in the body.
Sir, then you have taken only one kind of protein, PSA. Have you tried with any other complex protein? And does it show
We
any variation?
we have worked with hemoglobin. We have worked with this albumin, both human and bovine,
Okay.
and we are also working with another protein which is very interesting and has suffered a lot because of this glycation is catalase. So impairment of catalase can be realized that it will impair the whole antioxidant defense system.
Okay.
So
Okay.
we
Thank
are
you.
also working on those proteins. Because of the lack of the time, I could not show my other. We have also worked with the nanoparticles, and that we have the paper is under the review in the Journal of Biomolecular Structure and Dynamics. So zinc nanoparticles and
Thank you, sir.
thank you. So basically, my aim was to just talk about the biochemistry of diabetes, which we generally don't talk, and what can be some simple assays which can be done in the lab also to quantify them, and basically. So
Are there any questions for the speaker? Okay, if there are no questions, and we have come to the end of the session also. On behalf of Christ Deemed to be University, I thank Dr. Ahmad Ali for taking time to share his research on glycation and some of his research results. Thank you, Dr. Ahmad Ali.
yes. Thank you, Dr.
Thank you.
Siddharthan. Thank you, Dr. Sandhan. I could not get a chance to, as usual, this is one of the drawback of this
Yeah,
online lecture where we cannot see
Each
and
other,
interact
yeah.
with each other, and that would have been definitely so.
yeah,
Most probably, some in the future we may be able to meet each other and interact with each other for all kind of academic collaborations also and this thing. So I'm thankful to all of you and your administration for
Yes, of
organizing
course.
this wonderful course, and hope this interaction goes. So
yes, we look forward to make more collaborations with
we
you and come here to discuss.
please convey my thanks to all the people involved in organizing this FDP.
Okay.
Yeah. Personally, I have interacted with Dr. Siddharthan and Santhan. So
Yeah.
I'll be mentioning your name, but there are many other people who are, and thanks for listening. This.
I must thank Dr. Sainthan for suggesting a good speaker for today's FDPX.
So it's all my pleasure indeed, sir, for being a part of this thing, and I'm thankful to Ahmad Ali sir also, so that he kindly accepted the invitation, and he has kindly given his talk. It was a great talk, and since I'm writing reports, lots of things I have written. I don't know how much what to include. So thank you so much, Ahmad sir, for accepting our invitation, sir. Thank you so much, sir.
Thank you again for giving me this opportunity to interact with you. and I'll be always definitely looking forward to more interaction with you.
Yes,
So,
yeah, we also look forward to
and
it. Thank you.
I'm giving an invitation for you to please do come to Mumbai University once we are
Yes.
through this situation.
Thank you, everyone. Thank you.
Thank you. Thank you.
Bye.
Thank you. Thank you. Thank you so much.